Hello, thank you for joining us today. I'm Carrie Burton, your host for today's webinar, and with me today is Sarah Scheidler and Lucy Granger to reveal the science of Halloween candy. Before we begin, please note that you are on mute and will remain in listen-only mode throughout the webinar. If you have any questions or comments, please submit them at any time during the session using the control panel on the right side. We will be answering as many questions as possible at the end of the presentation, and we are recording today's webinar and will be sending the replay to you via email. And now I'd like to welcome Sarah Scheidler, Raman Application Scientist at Renishaw Inc. in the Spectroscopy Products Division. Hey, Sarah.
Hi everyone. Thanks for joining us for today's special Halloween-themed webinar. My name is Sarah Scheidler, and I'm an application scientist based out of our U.S. office. Our goal for today's webinar is to discuss some real challenges in food science and how Raman spectroscopy can be applied to those types of materials, but to have some fun with it and to look at some more sweet and potentially frightening samples. Please feel free to send any questions that you might have as you think of them during the talk. You can do that using the questions panel, and Lucy and I will try to answer them during the question and answer portion that follows this talk. Today we'll walk through how one might use Raman spectroscopy in the analysis of food products by looking at a couple of fun-sized examples. First, we'll use Raman imaging to look at samples of chocolate, dark, milk, and white chocolate. Then we'll use particle analysis to investigate JELL-O powder. And then we'll finish up with a couple of examples of hard candy and gum. A benefit of Raman spectroscopy is that it can be used to produce a chemical image of a surface. From this, we can get the distribution and particle sizing information of the different materials and chemistries present. This can also be helpful when looking for a small amount of a contaminant, such as in the trace detection experiment, where the high sensitivity of the Raman image can find trace materials that a bulk experimental technique might miss. The Raman image can also be used qualitatively and quantitatively to compare between different samples or batches to investigate any differences that are observed. Raman is a non-contact and non-destructive technique, which makes it complementary to other analytical laboratory techniques. And you could even do a Raman measurement on a sample and then follow it by a destructive technique. It is also very flexible as to what types of samples you can look at. Solids and solutions are both no problem. As long as you can focus your microscope onto the sample, you can collect a Raman spectrum. When we conduct a Raman spectroscopy experiment, what we get out is a Raman spectrum. And here, I've shown an example of a spectrum from a milk chocolate sample. The fingerprint region, out to around 2,000 wave numbers, can be used for the identification of the materials and the polymorphic form present in the sample. And in this spectrum, we can also see the CH stretching bands out at around 2,900 wave numbers, which can also be helpful in probing the solid-liquid phase transitions for polymorphic forms. In general, the Raman spectrum provides a wealth of information about the sample's surface. By looking at different parameters, we can derive different information about the sample. For instance, by looking at the frequency of the characteristic bands that are occurring, we can identify the chemistry and materials present. For example, with a chocolate sample, we can differentiate between ingredients such as lactose and sucrose. We can also look at how the width of a particular band varies across the sample to see changes in the crystallinity, which gives an idea about the order and disorder within the sample. Our first type of treat that we're going to look at is that Halloween classic, chocolate. Specifically for chocolate, we can use Raman to identify the materials that are present. So down below, I'm showing 3 reference spectra for materials that might be present in a chocolate, such as fat, lactose, and sucrose. So you'll notice that all of them have slightly different spectra. We have different characteristic peaks that we could use to identify where these materials are located within our sample. We could then use a Raman image to look at how these materials are distributed spatially, and we can even analyze the different particle sizes of the ingredients. We could also look at the structure or different polymorphs that are present throughout different stages of the food production and storage. So we could look at how this chocolate is aging over time. We'll start by looking at milk chocolate. So milk chocolate is prone to a high fluorescence background, but we can avoid that fluorescence by using different excitation wavelengths. For example, 785 nanometer excitation, which is shown in blue, and 830 nanometers, which we've shown in red. After baseline subtraction, we can see Raman bands that are characteristic of sucrose with the band at 850 wave numbers, cocoa butter with the bands that are around 1450 wave numbers, and lactose with the band at 1086 wave numbers. We can use these different bands to identify the materials and their distribution in a Raman image of this sample. Now we'll look at an example of bloom chocolate. So you've probably seen bloom chocolate before. Usually it's when you go to eat an old piece of chocolate and you notice that it has white areas on the surface of the chocolate. While not harmful, it can have an unappealing look that might make you think twice about eating it. We then found an area of our milk chocolate sample that had bloomed, and we went and investigated what materials were present in that bloom. And so now if we were to look at the Raman image, what we see in red is the fat, and that is what is at the bloom location. And then we see in green lactose and in blue sucrose. So we can then determine that that chocolate bloom was made up of different fat crystals. By looking at the Raman spectrum that was collected at a point within the chocolate fat bloom, we can use the number of peaks that we're seeing and also their peak position, and that's going to give us an idea of the different polymorphs of the fat that are there. And we can actually relate the CH twisting mode to the degree of coupling between the alkyl chains, and this will give us, again, some idea of the crystallinity present within the fat bloom. Within this Raman image, if we were to only look at areas that correspond to the fat, we can plot the intensity of the Raman peak at 1300 and compare that to the peak at 1440. And this shows us that the fat areas that are within the bloom, that we have more of this presence of the peak at 1300, which is related and attributed to the CH twisting. And we also see that we could look at the relative peak position of that CH twisting peak, and that we see a shift towards a lower wave number in the fat bloomed crystals. And this is going to indicate a more crystalline structure within that fat bloom compared to the other fat molecules found in the sample. Now, while chocolate of any sort seems like a real treat to a spectroscopist, chocolate can actually prove to be very tricky, especially with the amount of fluorescence that is observed with dark chocolate. We can see this in the raw data that was collected on the dark chocolate sample, which is shown in the figure on the left. We see the high fluorescence baselines present with 785 nanometers, making it hard to observe the Raman bands, which are of interest. When we baseline subtract the data, which is shown on the right, the Raman bands are more clear, but we do see a great benefit with being able to switch to a different excitation source to 830 nanometers for this measurement. We can then clearly see the Raman bands for sucrose at 850 wave numbers and the CH deformation bands for the cocoa butter around 1450 wave numbers. Now we move on to imaging of white chocolate. First, we snapped a white light image of our sample and defined our mapping area. On the right, we see our Raman image, where we can see the sucrose and lactose particles are distributed within a matrix of fat. Every pixel in this map contains a full Raman spectrum, and we compared those to the references for the ingredients to come up with this image showing the distribution of each material. Using this image, we can start to identify how the distribution of particles within the fat might impact the taste or texture of the chocolate and also how stable the chocolate is. This is a larger area map over a Hershey Hug chocolate containing just under 1 million spectra. Again, we see the distribution of the lactose and sucrose in the fat matrix. Now, this type of imaging could be used for quality control to check the distribution of the ingredients, and if there was some issue with either mouthfeel or the texture of the chocolate, Raman could be used to probe why these changes might be occurring. As any baker knows, the thought of melting and tempering chocolate is sure to strike fear in the hearts of many a baker. Chocolate is very sensitive to the temperature as you are melting it, and it can burn, and any added moisture can cause the chocolate to seize, turning your beautiful chocolate into a nasty, clumpy mess, which will not continue melting, and while it still tastes fine, has a terrible texture, not only on your dessert, but also when you eat it. As someone who enjoys baking myself, this phenomenon was both annoying to me, while also potentially interesting from a ramen perspective. We decided to look at samples of white chocolate in chip form, melted, and then seized to see if ramen could reveal what was happening during this process. Here we start with our white chocolate chips, straight from the bag. We collected a ramen map on an area of the surface of the chip and generated a component analysis map looking for the three main ingredients that we've talked about previously: fat in red, lactose colored green, and sucrose colored blue. Compared to some of the earlier images we've looked at of other white chocolate, we see that the fat and sucrose are spread out fairly evenly throughout the chocolate chip, and we see very, very small areas of lactose. I've colored them green and circled them also with green to make them easier for you to see. Next, we took some of those chocolate chips and melted them and spread it into a layer onto a stainless steel slide. In the ramen image, we again see a fairly uniform distribution of the sucrose within the fat. Now onto our seized chocolate. Even from just looking at the seized chocolate on the right side of that slide, we can see that it's very clumpy and didn't spread nicely on the slide. And now from the ramen image, we can start to see why. Whereas in our chocolate chip and the melted chocolate samples, we had a uniform distribution of the fat and sucrose. In the seized chocolate, we can see areas of the fat and sucrose have separated out, and we see large areas of the sucrose banded together. Interestingly, we also see larger particles of the lactose as well in this seized chocolate. As far as Halloween party treats and goodies go, nothing seems quite as eerie as a bowl of JELL-O or a JELL-O mold seeming to be wiggling all on its own. Here we will talk about JELL-O powder before it has been made into JELL-O, and we will analyze it using our particle analysis software. Here we have our JELL-O powder, and we see its main components of sugar, gelatin, and some acids and other ingredients for flavor and color. We sprinkled some of it onto a stainless steel slide and imaged it using dark field illumination. The dark field capability provides a really nice contrast and allows us to clearly distinguish the powder particles from the background of the slide. We then input this image into our wire particle analysis application and had it identify the particle locations. We have the ability of changing how it's doing the binning of the image to try to separate the powders further, but I think this image is separated really well, and we can see the individual particles as the software has identified them. So now I'm going to show you how we can use that particle analysis software to analyze this JELL-O powder. So right now I have a list of all of those particles that were identified in the previous image, and I have them sorted by their equivalent circle diameter, and I'm showing all particles that have a diameter greater than 3 microns. And I can go through this list and click on different particles, and it's going to show me which particle I've selected over in my image. So I can go ahead and look at different size particles. Right now I'm moving to some of the smaller ones, the 3 micron particles, and I can see each of the particles when I click on it. And I can sort them by different parameters as well, their position, their area, and I can come up with a list of which particles I want to do further analysis on. So in this case, I'm going to highlight all of my particles in my image, and I'm going to tell it that I would like to look at only 50%. I want it to pick a random 50% for me, and those are the ones that I'm going to put into a box for my particle analysis. So now I'm just going to look only at my boxed particles, and I can see a list of just my particles that I'm going to analyze. And I can scroll through and look at the different sizes of those particles. Now I'm going to go ahead and tell the particle analysis software what type of measurement I would like to do. So I'm going to open up our data collection tab. Here I have the option of picking a different template. I can change the power. I can also change the exposure time, how many accumulations I want to do. And then when I'm all happy, I press run. Okay, so we're going to start collecting our data from each particle. So what the software is doing is it's moving to each particle location and collecting a spectrum. I have live track turned on for this measurement, so I'm going to switch over to wire, and we can actually see that the height is automatically adjusting to account for differences in the particle height at each location. And so now we can just see our data as it's starting to accumulate in the software. In this case, I'm going to be analyzing over 2,000 particles, and Wire is going to estimate that the completion time is going to be just under 2 hours to analyze all of those particles. Now Wire is done collecting all of our spectra, and we're going to be able to look at all of our data. So in this, I'm clicking on different particles, and I'm seeing the spectra that was collected at that location. I can also click on a particular particle, and to do processing of this, I can either transfer all of the spectra to Wire, or I can run what's called a chain, and that's going to be an automated data processing. So in this case, I'm going to baseline subtract all of these spectra, and then I'm going to have it do a library search to identify the material present at that particle location. Okay, so let's go ahead and analyze these results. We can click on any of our particles in our list. We can sort them by material type. Here I'm picking all of my adipic acid. I can click on a particle in the image, and it's going to bring up the spectrum and the library search that was conducted for that particle. I can also now start to tabulate my results. And here I'm going to look at the distribution of the different materials by the particle area. So I can click on the gelatin particles and see how many particles I have and what size they are. I can look at some of my flavorings. Really I can tabulate these results, and I can customize them to give the information that I'm most interested in looking at. Let's look at the image of the particles colored by material name again. Here we can see that the large particles are primarily our sugar crystals, while our medium-sized particles are mainly gelatin and adipic acid, which is both a flavorant and a gelling aid. And the smaller particles are the adipic acid, citric acid, sugar, and other colorings. In the particle analysis software, we could filter our particles based on size, for instance, and only analyze the smaller particles if that's what we're interested. We can then create images like this, where we've color-coded our particles by their different material identifications. We also took some of our powder and imaged it using Raman chemical imaging. Here we can again see the sugar crystals make up the majority of our surface area, and we also see gelatin, adipic acid, and citric acid, including some smaller particles on those big sucrose crystals. We used an NVIDIA contour microscope for this imaging with a live track capability in order to maintain focus on the sample, even though the height was varying over 400 microns. And we can visualize this height data in the image for this on the right. We can also use this height data to display our ramen as a 3D surface. So I have a quick video where I'm going to change it into a 3D view. And now we can see our particles as their crystalline material and the varying heights. We can zoom in. We can actually click on each pixel and we'll see the spectrum that was collected at that location. But really we just get a feel for what our surface actually looks like with this live track capability. We also had no loss in signal intensity throughout our measurement because our sample was always in focus. From the Raman image, we can also estimate the concentration of the various ingredients. While particle analysis allowed us to measure over 700 particles at 1 second acquisition each, which allowed us to reduce the measurement time, the Raman imaging allowed us to visualize the complex geometry of this powdered surface. Now we'll move into our final examples, because what would a Halloween webinar be without some discussion of pure sugar candy? For our candy example, we are going to take a look at a sweet tart, conversation heart, and analyze one of the ingredients using particle statistics. On the left, we see a white light image, which we use to collect a Raman image over the whole heart surface. Again, we were using live track on our contour system to maintain focus and adjust to the varying height of the heart surface. This meant that no sample prep was required. We could just put the sample under the microscope and start collecting our image with no loss of signal. We used the ingredients list from the candy box as a guide for our library searching. We can see the dextrose, maltodextrin sweeteners, which give the sweet tart that sweet flavor, which we've colored here in pink, as well as particles of malic acid, which are white, that give the candy that tart flavor. We do see some additional chemistry in yellow, which we need to do additional library searching on to be able to identify. And just to give an idea of the type of quantification that you are now able to do on this Raman image, I took those malic acid particles, which were white in our Raman image, and did an image analysis on just those malic acid particles. And the results that we get back are we can look at the number of particles, we get distribution of the size, we can determine how much of the percentage area that these malic acid particles covered, and then we can also look at the size distribution of these malic acid particles. If we were to click on any single particle, we could get information about that one, how big it was, what the equivalent circle diameter would be, etc. And we could use all of this to quantify how much of the material was present and where it was located within our sample. We've also looked at some other food products, including bubble gum. So we collected a Raman image on the surface of a piece of bubble gum, and what we see here is that the majority of it is sugar in red and polyethylene covinyl acetate in blue. We also see malic acid in cyan, citric acid in magenta, and then talc in white and calcite in yellow. And those are actually added to the gum to prevent it from adhering to the packaging. This was, as you can imagine, a piece of gum is not a smooth surface, so there is some curvature to the piece of gum that we looked at. We have the option of doing what we call a live track measurement. So the sample height is actually changed throughout the measurement to keep the sample always in focus. And so what we've shown here is the height information displayed with that Raman image. So you kind of get a 3D view of what the surface of your piece of gum looks like. All of the information that's present in these images can actually be quantified. So we could look at the percentage coverage, the domain sizes for the different ingredients, and then we could also compare them between samples to see if there was any variation within different pieces of gum. So to conclude, we've shown that we can use Raman imaging to look at a number of different Halloween treats. We can use Raman to identify the chemistry and the distribution of key ingredients within samples such as chocolate, gelatin powder, bubblegum, a sugar candy. And by using Raman imaging, we're able to characterize different polymorphs of ingredients, such as fat within a chocolate sample, and use that as a way of looking at the distribution of ingredients, such as sucrose and fat that are within seized chocolate. We also looked at powdered samples, such as our gelatin powder example, and we can see that they readily lend themselves to measurement using particle analysis. In those cases, we collect a Raman spectrum from each particle, and we can quantify the chemistry that's present along with the different particle sizing information. We're also able to use Raman imaging to quantify the particle size and distribution of ingredients within a sample. And for this, we looked at a sweet tart example, and we could see the malic acid, which provides that tart flavor of the sugar candy, was found in the Raman image. And then we were able to use particle statistics to look at how the malic acid was distributed and sized within the candy. And just overall, in general, we found that being able to use a contour system with LiveTrack was really beneficial for these food samples because they're fairly rough and uneven. And so with the automatic height adjustment, our samples always remained in focus, and we could reduce the amount of sample preparation needed, and also it allowed us to create really nice 3D images where we're able to see really what the surface and the morphology of these samples look like. Thanks for listening. I'm now going to invite Lucy Granger, a fellow applications engineer, to join me for the question and answer period.
Thank you, Sarah. And I'd like to note that we will be sending a link to the recording in case anybody would like to review it again. So we'll probably get that out tomorrow or Friday. And if you have any questions, please submit them now. We'll have a few minutes to answer some of them. So please welcome Sarah again and Lucy. Hey guys, or I should say girls.
Right, thank you, Sarah. That was really interesting. I think we've got some good questions coming through. So I'm going to start off the first one. So which model of Renishaw Raman system is being used here?
Sure. So all of the data that we presented today was collected on our NVIDIA contour that's actually sitting behind me. But there's no reason why you couldn't do it on any NVIDIA system or even your RA802, which also has the live track capability.
Okay. Got another question here. So awesome presentation. That's not me, that's the questioner. Does using LiveTrack to maintain focus slow down the scanning process of acquiring Raman maps in any way? And also, I suppose this would be a good opportunity for you to describe a little bit more about what LiveTrack is and how it works.
Sure, yeah. So we can start by talking a little bit about LiveTrack. So LiveTrack is just going to automatically adjust the height of the stage as you're scanning your sample so that your sample is always going to remain in focus throughout the measurement. So even if your sample has like a rough surface or a curvature to it, LiveTrack will adjust the stage so that it's always in focus. And no, it does not slow down your acquisition time. That's still going to be determined by you when you're inputting your parameters for your measurement. And so it's all built in and it doesn't add any additional analysis time. And it doesn't require you to know your sample beforehand and predefine that surface. It's an automatic, as you're collecting your data, the height is adjusting to maintain focus.
Okay. So in the particle analysis software, what's the minimum particle size that it can select and probe?
Sure. So that's a really good question, and it's going to depend on a number of different factors, and we can kind of talk about what those would be. So as far as your Raman measurement itself and how good of spatial resolution you can obtain. That's going to be dependent on the wavelength of light that you're using and also the objective, but we can get down to sub-micron. So if you have a feature that's less than a micron, that's no problem, and we would be able to collect a Raman spectrum from it. Now, as far as the particle analysis software and the particle size that you're able to look at, it's going to depend kind of on how big your samples are and what the features are that you're trying to determine. So for instance, with the gelatin powder that you can actually see in the image behind me, those crystals were pretty big. So I was just able to image them with a 5x magnification objective and be able to use that image to identify my particle locations. Now, if I was trying to find maybe a 1 micron particle on a large filter, I'm going to need to image it with a little bit higher resolution. So maybe I'm going to use a 20x objective to collect my image to be able to identify where those particles are located. And then for my data, my Raman data acquisition, I would switch to a higher magnification objective to actually collect that data. So yes, you can analyze, particles down to a micron. It just really depends on what you're looking for with how you want to set up your experiment.
Okay. So along those lines, how deep can it go? So I assume, how deep can we analyze into the sample?
So Raman's a fairly surface sensitive technique, and it's going to depend on how far the laser is penetrating into the sample. So it's going to depend on characteristics of the sample itself and how transparent it is to how deep you'd be able to go. But you are able to do depth profiling measurements or collect a 3D volume image of your sample. So that was something that I actually wanted to look at, was now take that JELL-O powder and make it into JELL-O and then do like a volume map. But I didn't get there in time for this talk, but it's definitely something that you could do depending on your sample.
Okay. We've got a few questions regarding library searching.
Okay.
So I suppose rather than me asking the individual ones, maybe you can talk a little bit around the libraries we have available. And
sure. So in this work, I used some of our available libraries. So we have a polymers library, an excipients library, which was really helpful for this. And you can use other external libraries as well through the wire software to be able to identify your materials. And then the other thing that was really helpful is that you can build up your own libraries. So if you have components that you know are in your sample, it's really best to take a reference spectrum from the pure material itself. So for the JELL-O example here, I actually bought some plain unflavored gelatin and collected a spectrum from that as well, so that I could compare it to what was in the mixed powder. So I built up my own library and then could do my library searching based on my own library that I had generated. But yes, we do have a number of libraries available with some common ingredients as well.
Okay. And a question specifically about the reflex system. Suppose we can stretch that out to all the systems available. Can a 3D map be obtained using the InVia reflex system?
Yes, I believe so.
Okay.
Yep. And so the only thing is that the contour is the system that has the live track capability, and that's what we were talking about with the auto-focusing. That was really helpful. But yes, you can do a 3D map with your reflex.
Okay. Got another question here about live track, just asking if it's available for any laser wavelength. So this person has UV and near IR lasers.
So it's definitely available for the 785 laser, which would be your near IR. And it's also available for the visible wavelengths. I would need to check on the UV system. I don't believe that it is available.
Okay. A question about the 802, RE802. Okay. How effective is the RE802 to detect 1% of particles in a formulation with a size of between 1 and 5 microns? So I suppose they're referring to maybe the API content?
Yeah, so if you're looking for a 1% particle within, let's say you have a tablet. So the 802 is our pharmaceutical analyzer, and it's really, it's designed to be really great at handling tablet samples. So you could load up a bunch of tablets in there and collect your data. So for that system, depending on your step size that you are using to analyze your tablet, you would definitely be able to look at 1% concentration of an API compared to everything else, because all you're looking for are specific pixels that are corresponding to that reference spectrum of the material that you're interested in. So my guess is that it would be able to measure that.
Okay. I think we've got time for maybe one or two more questions.
Okay.
So we've got one here. If you don't know the kinds of chemicals that will be in a sample like candy, could you have alternatively run a principal components analysis? How could you optimize your use of PCA to make sure the principal components correspond to pure substances as if you'd gotten reference spectra?
Sure, yeah, so that's a great question. So I mentioned it in here that when I was collecting these candy images, I cheated and I looked at the box and for the ingredients and used them to help me generate my maps. But you're right, a number of times you don't really know what's in the sample that you're analyzing, and so maybe you don't have reference spectra that you could pull. So in those cases, you would want to do a multivariate technique that would be an unconstrained model. So a principal component analysis, or we have another one that's an empty modeling technique. And those are going to look for trends in your data set itself and identify what components might be present. So you could definitely use principal component analysis to try to pull out the ingredients that would be present. And what that would do is identify similar areas in your map that all had similar spectra. You could then look and compare those spectra to a library and try to identify what components were present. So yes, you definitely could use other techniques to identify what was in those samples.
Okay, one final question then. Is the user able to generate their own libraries?
Yeah, so that's a great question, and absolutely. And we recommend that if you have components in your samples that are not in libraries, it's really easy. You would just collect your spectrum and then save it to your own library that would be available for your local searching.
Okay, perfect. Well, thank you very much, Sarah.
Yes, thank you so much. And if we didn't get to your question, we will be having some follow up with you directly to answer those questions. So thank you again for joining us today. We hope you can join us for our next webinar, which will be with Farm Tech. And if you'd like to learn more or have any additional questions, please contact us via email. And please follow us on social media so that you can stay up to date for all of our upcoming events. Thank you so much, and we hope you have a great Halloween.
Bye, thank you.